Hi there. Welcome to the channel. Today we are going to learn about DNA. Now this is a very interesting topic because you can use this knowledge in real life. So let's get started. But before that, I have a small quiz for you. In humans, DNA is located in which of these? A. Nucleus, B. Cytoplasm, or C. Vacuole? Well, the correct answer is A. Nucleus. And the other options are incorrect. DNA is present in the nucleus of the cell. This molecule carries all the genetic information in an organism. It is a very long, thin molecule which is folded in such a way that it can fit inside the nucleus of a cell. Now, if it was unfolded, it would be more than 2 meters in length. Have you ever wondered what is DNA made up of? To answer that question, let's do this activity. Take 2 long sugar sticks and 2 long plastic straws. Twist both the sticks together and then use tape to make a model of a ladder like this. Now this activity shows how 2 long strands of DNA are twisted around each other to form a ladder-like structure called a double helix. Now let's look at the components of DNA. Deoxyribonucleic acid, or DNA, is made up of 3 components: a sugar, a phosphate group, and a nitrogenous base. These components are attached to each other in a specific order. The sugar and phosphate groups alternate and form the backbone of DNA. They are linked together by strong covalent bonds. The nitrogenous bases extend inward from the backbone and form hydrogen bonds with bases on the opposite strand. The sequence of these nitrogenous bases is what determines the genetic information of DNA. Now let's look into the structure of DNA in detail. Each component of DNA plays a significant role in the functioning of this molecule. Let's first look at the sugar. The sugar in DNA is called deoxyribose. It is a 5-carbon sugar that forms the backbone of the DNA molecule along with the phosphate group. The carbon atoms in the sugar are numbered from 1 to 5. The nitrogenous base is attached to the first carbon. The 5' carbon of one sugar molecule forms a bond with the phosphate group to connect to the next sugar molecule. Now let's look at the phosphate group. In DNA, the phosphate group is attached to the 3rd carbon of one sugar and the 5' carbon of another sugar. The phosphate group negatively charges the DNA molecule and makes it acidic. It also gives DNA its hydrophilic nature, or the ability to attract water molecules. Finally, let's look at the nitrogenous bases. There are 5 types of nitrogenous bases found in nature. 2 of them, adenine and guanine, are purines. The other 3, cytosine, thymine, and uracil, are pyrimidines. Uracil is found only in RNA, so we will focus on thymine, cytosine, and adenine and guanine here. A purine base always pairs with a pyrimidine base. Adenine always pairs with thymine through 2 hydrogen bonds, whereas guanine always pairs with cytosine through 3 hydrogen bonds. Let's now look at the structure of a nucleotide. Nucleotide is the building block of DNA. It has 3 components: a nitrogenous base, 5-carbon sugar, and a phosphate group. When a phosphate group is bonded to a sugar molecule, it forms a nucleoside. When a nitrogenous base is added to a nucleoside, it forms a nucleotide. For example, if adenine is added to the nucleoside adenosine, it forms the nucleotide adenylic acid, or AMP. Now that we know the components of DNA, let's see how they combine to form a DNA molecule. As we discussed earlier, nucleotides are the building blocks of DNA. They are joined together by phosphodester bonds. These bonds are formed between the phosphate group of one nucleotide and the 3' carbon of the next nucleotide. This forms a long chain of nucleotides. The sequence of nitrogenous bases along this chain is what carries the genetic information of an organism. The 5' end of a DNA strand has a phosphate group, and the 3' end has a hydroxyl group. You might be wondering, what does this anti-parallel arrangement mean? Let me explain. The 2 strands of DNA run in opposite directions. One strand runs from the 5' end to the 3' end, and the other strand runs from the 3' end to the 5' end. This is called anti-parallel arrangement. The anti-parallel arrangement of DNA strands is important for DNA replication and protein synthesis. During DNA replication, the 2 strands of DNA separate, and each strand serves as a template for the synthesis of a new strand. The anti-parallel arrangement ensures that the new strands are complementary to the template strands. During protein synthesis, the DNA molecule unwinds, and the genetic information is transferred from the DNA to a messenger RNA molecule. Again, The anti-parallel arrangement of the DNA strands is important for this process as well. Another interesting fact about DNA is that it is a very stable molecule. This is due to the strong covalent bonds that hold the sugar-phosphate backbone together. The hydrogen bonds between the nitrogenous bases are relatively weak, but they are important for DNA replication and protein synthesis. The stability of DNA is important for the proper functioning of cells. If DNA were too unstable, it could break down and the genetic information it carries could be lost. Now let's see why DNA is the molecule of heredity. The ability of DNA to replicate itself is the basis of heredity. During DNA replication, the two strands of DNA separate, and each strand serves as a template for the synthesis of a new strand. The new strands are complementary to the template strands, which ensures that the genetic information is passed on accurately. Now let's do an activity to understand DNA replication. You will need 2 long sticks, tape, paper clips, string. Take the stick and wrap the string around it to make a model of a DNA strand. Use paper clips to represent nucleotides. Tape the ends of the string together to form a circle. This model represents a DNA molecule. Now pull the 2 ends of the string apart to separate the 2 strands of DNA. Each strand can serve as a template for the synthesis of a new strand. Add paper clips to the single strands to represent nucleotides. The new strands are complementary to the template strands. In this activity, the stick represents the sugar-phosphate backbone of DNA, the string represents the nucleotides, and the paper clips represent the nitrogenous bases. This activity shows how DNA replicates itself during cell division. The enzyme helicase unwinds the DNA molecule, and the enzyme DNA polymerase synthesizes new strands that are complementary to the template strands. DNA is the molecule of heredity, because it can pass on genetic information from one generation to the next. The ability of DNA to replicate itself is the basis of heredity. Now that we know about the structure and function of DNA, let's move on to the next topic. If you want to become a member of our community, click on the link below. In eukaryotes, DNA is organized into structures called chromosomes. Chromosomes are thread-like structures located in the nucleus of eukaryotic cells. They carry the genetic information of the organism. Each chromosome is made up of a single molecule of DNA that is folded and condensed. Eukaryotic DNA is linear. Each chromosome has a centromere, which is the region where the 2 sister chromatids are joined. The centromere also plays a role in the separation of chromosomes during cell division. The number of chromosomes in eukaryotic cells varies. For example, humans have 46 chromosomes in their somatic cells. Now let's see how DNA is packaged into chromosomes. As we discussed earlier, DNA is a very long molecule that must be folded and condensed to fit inside the nucleus of a cell. This is achieved through a series of folding and packaging processes. DNA is first folded into a structure called a nucleosome. A nucleosome consists of DNA wrapped around a core of histone proteins. The DNA wraps around the histone core approximately 1.65 times. The nucleosomes are then folded into a 30-nanometer fiber. This fiber is further compacted to form loops and domains within the chromosome. Finally, the chromosome is condensed to its most compact form during cell division. The packaging of DNA into chromosomes is important for several reasons. It allows for the efficient organization of genetic material within the nucleus. It also helps to regulate gene expression by controlling the accessibility of DNA to RNA polymerase. Additionally, the packaging of DNA into chromosomes is essential for the proper segregation of genetic material during cell division. Now let's move on to the next topic. The central dogma of molecular biology describes the flow of genetic information in a cell. It states that genetic information flows from DNA to RNA to protein. DNA is the molecule of heredity, carries the genetic information of an organism. During transcription, the genetic information in DNA is transferred to a messenger RNA molecule. In eukaryotes, this process occurs in the nucleus. During translation, the mRNA molecule is used to synthesize a protein. This process occurs in the cytoplasm. So this brings us to the end of this topic. We hope that you have enjoyed learning about DNA. We will be back soon with more interesting topics. So stay tuned and keep learning. Goodbye.